Dear students, Assalamu alaikum. I hope you and all are well by the grace of Almighty Allah. In pandemic situation of COVID-19, please follow the rules of World Health Organization, that means WHO, about COVID-19, such as avoid gathering, maintenance, social distance, washing hands frequently with any kinds of soap. Today we learn about biology second paper with online multimedia presentation. So let's go. We will start our presentation. So dear student, here I am Shahabuddin, lecturer, Department of Biology, Milestone College, Uttara. Today's lesson, today we will discuss about chapter 9, the name continuation of human life. So you are watching the image, and here the continuation of human life is seen by the image. Today's the topics that I discussed, first of all, the process of implantation. Second one, the process of human embryo development, or the process of development of human embryo, or embryogenesis. First of all, learning output, that means what will we should learn from this lesson. Number one, we should be able to learn about the definition and process of implantation, and we should be able to learn about the process of formation of human embryo. So first of all, we have to know what is implantation. Implantation is a process in which the embryo adheres to the wall of endometrium of the uterus in the very early stage of pregnancy is called implantation. At this stage of prenatal development, the embryo is a blastocyst. About 7 days after fertilization, the blastocyst attaches itself to the uterine endometrium and burrows into it. The implantation site is usually on dorsal wall of the uterus. Here some the guideline or some stages of process of implantation. First of all, here zygote. What is zygote? Zygote is the first step of life, which is formed or consists of the fusion of sperm and ovum. That means male and female gamete. Here zygote divided through the process of mitosis cell division is called cleavage and produces blastomere. About 3 days after fertilization, a ball of 12 or more blastomeres called a morula enters the uterus. A cavity soon forms in the morula covering into a blastocyst. Here blastomere form blastocyst with the process of morula. And here the figure of blastocyst. Blastocyst consists of 3 parts. One is trophoblast, another one is blastocele, and third one is embryoblast. Here an inner cell's mass or embryoblast, which gives rise to the embryo. Blastocele. Blastocele is a cavity of blastocyst. Dear students, I repeat this. Blastocele is a cavity which is located in the inner side of blastocyst. And trophoblast. Trophoblast is the thin outer layer of blastocyst. After 4 to 5 days after fertilization, the zona pellucida disappears and blastocyst attached with the endometrial epithelial. It takes 6 to 9 days to complete the whole process of implantation, and other sublying cells proliferate rapidly, forming the placenta and the various membranes of pregnancy. So dear students, this is the process of implantation. This is so much important for us. It can appears our exam as a knowledge based question or the comprehension based question, and also application based question. Here's some information for you. Cleavage, that means how to blastocyst consist from the zygote. Here the first cleavage produces two daughter cells called blastomeres. Morula, the 16 or more cell stage. By the 4th or 5th day, the pre-embryo consists of 100 or more cells. Blastocyst, a fluid-filled hollow sphere composed of a single layer of trophoblast. Dear students, I have talked about this in the last slide, so you can watch again and again about this process. The next topics of our lesson: development of human embryo. Development is a word which can indicate the development or developed of any kinds of substances or any kinds of processes, any kinds of things, etc. In biology, development is a progression from zygote to child or larva of any kinds of animals or human. Dear students, I can repeat. Development is a progression from zygote to child or larva of animals or human. Then another keyword, embryogenesis. Embryogenesis, the process by which the embryo is formed from the zygote is called embryogenesis. Here two Latin words are take part to consist this word embryogenesis, embryo and genesis. Embryo means the developed part of zygote, and genesis from means to produce. Genesis means to produce. That's why embryogenesis is called the process by which the embryo is formed from the zygote is called embryogenesis. And another word, embryology, is the physical branches of biology. Embryology is the physical branches of biology which deals about the development and the processes of embryo formation. Here 3 stages of development of human embryo. Number 1, cleavage. Number 2, gastrulation. And number 3, organogenesis. First of all, I talk to you about cleavage. Here zygote is formed by the fusion of male and female gamete, that means sperm and ovum. So cell division with no significant growth, producing a cluster of cells that is same size as the original zygote is called cleavage. Dear student, cell division with no significant growth, producing a cluster of cells that is the same size as the original zygote is called cleavage. At least 4 initial cell divisions occur, resulting in a dense ball of at least 16 cells called the morula. The different cells derived from cleavage up to blastula stage are called blastomere. Dear students, what is blastomere? Blastomere is the different types of cells which is derived from cleavage at the stage of blastula. After the cleavage has produced over 100 cells, the embryo is called blastula. The blastula, usually a spherical layer of cells, is called blastoderm. Here blastula is consist of two parts. Number one, blastoderm, and another one, blastocele. So blastoderm is a spherical layer of cells, and blastocele is the cavity of blastula. Next on gastrulation. Gastrulation is a phase early in the embryonic development of most animals, during which the single layered blastula is recognized into a trilaminar structure known as gastrula. During gastrulation, cells migrate to the interior of blastula, consequently forming three germ layers. Dear students, please watch this figure or notice the following figure. Here, three germ layers are known as ectoderm, endoderm, and mesoderm. The outer layer is called ectoderm, the middle layer is called mesoderm, and the inner layer is called endoderm. Dear students, those animals contains these three types of germ layer, those are called triploblastic. Those are called triploblastic animal, and who have not contained mesoderm, those are called diploblastic animal. Okay. Then archenteron and blastopore. The internal fluid filled cavity of gastrula is known as archenteron. Dear student, I repeat this. The internal fluid filled cavity of gastrula is known as, this is one types of cavity is known as archenteron. It opens through a pore, is called blastopore. Then the process of organogenesis. There is student here two Latin word. One is organ and another one is genesis. Organ means a group of tissues which is originated from a same place and perform a special function is called organ, and genesis means to produce. So organogenesis is a process by which the ectoderm, endoderm, and mesoderm develop into internal organs. Organogenesis, I repeat this, organogenesis is the process by which the ectoderm, endoderm, and mesoderm develop into the internal organs of organism. Internal organs initiative development in humans within the 3rd to 8th weeks in uterus. A germ layer or its parts do not predetermine to form a definite organ. Primarily, these germ layers have ability to different functions, but this power brings to an end within a definite period. Cells of a definite germ layer when begin to form a definite structure. So, organogenesis continues until the definite characteristics of the organ are achieved. So, dear students, this is the process of human embryonic development, or this is the different stages of development on human embryo. Today's question, here some question from today's lesson. That means evaluation from today's class or today's lesson. Here some knowledge based questions are available from these topics: implantation, development, embryology, cleavage, gastrula, organogenesis, etc. Here some comprehension based question: what does mean by implantation? What does mean by cleavage? What does it mean by organogenesis, etc. And the application and higher ability based question from today's topics is first one, describe the process of implantation and describe the process of development of human embryo with label diagram. So dear students, I hope you are feeling well to do this class. If you faces any kinds of problem from this class, you will contact your subject teacher and solve the problem that are creating problem to you. So thanks to all. Allah Hafiz.